Jai Hind, dear students. I am Dr. Preeti Mittal and today we are going to start with the chapter Cell, the fundamental unit of life. A cell is known as a structural and functional unit of an organism. But why is it called so? A cell is known as a fundamental unit because a cell has a capability of performing all the functions that are performed by an organism. For example, respiration. Each and every cell of our body performs respiration, digestion of food, take up of food, then utilizing that food, apart from that, excreting out the waste. So, all the functions that are performed by an organism are performed by each and every cell of that particular organism. And that is why the cell is known as a basic structural and functional unit of an organism. Apart from that, there are some cells which have the capability of existing as independent cells. That is present in the unicellular organisms. Means a unicellular organism like amoeba can perform all its function, carry out its entire life just with a single cell that it is having. Okay, so we'll read about all that, but let's start with basic things like who discovered the first cell. So, the first cell was discovered by Robert Hooke. But Robert Hooke did that with a very simple microscope. And the cell that he discovered was a dead cell because that was from the cork. Cork is something that is found in the bark of tree. So, the bark of tree, the outermost portion of the tree. So, that is made up of dead cells. And that is having the cork. So, from that cork, the first cell was observed for the first time by Robert Hooke. And he only gave the term as cell. In Latin, cell means a small room, a little room. So, this term cell was also given by Robert Hooke. Then, Robert Hooke, he discovered the cell that was by his self-made microscope. So, he constructed a microscope on his own and just observed the cell. So, isn't it amazing? Then, the first living cell was observed by Leeuwenhoek. Anton von Leeuwenhoek was a scientist who observed the first living cell in pond water. He observed that cell. Okay. Then, Purkinje scientist was there who coined the term protoplasm. Now, what is protoplasm? Protoplasm means the nucleus that is present inside a cell and the cell cytoplasm. So, nucleus and cytoplasm of the cell together make up the protoplasm. So, this term was coined by Purkinje. Apart from that, who discovered the nucleus? So, the discovery of nucleus was made by Robert Brown. Remember, the first cell was Robert Hooke and the nucleus inside the cell was discovered by Robert Brown. Okay. Then, Schleiden and Schwann were two scientists who gave the cell theory. They told us something about the cell and that cell and that whatever they told, that is known as cell theory. So, remember that Schleiden and Schwann were two botanists who gave us the cell theory. But the cell theory was further explained by Virchow. Okay. So, what does the cell theory states? According to the cell theory, all the organisms, whether it is unicellular or multicellular, is made up of cells. It could be a single cell in case of unicellular organisms. It could be many cells in case of multicellular organism. Okay. So, each and every organism is made up of cells. Second point is that each and every organism starts its life journey as a single cell. So, every organism, whether it's human beings or it's plants, that all starts their journey as a single cell. And third point, every cell arises from the pre-existing cells. So, if a new cell has to be formed, that will be formed from some pre-existing cells. Okay. Next was that every organism This cell is a basic structural and functional unit of an organism. Means every cell is capable of doing the basic functions that an organism performs. So this is all the 4 points of the cell theory. Now, these cells could be, the organisms could be divided as unicellular or multicellular based on the number of cells that they have in their body. An organism, if it is having a single cell only, that is known as unicellular organism. But if an organism is having many cells like plants, human beings, dog, cat, elephant, tiger, whatever we see around us, all the higher animals, they are all known as multicellular organisms because they are made up of more than one cell. So the organisms could be classified as multicellular or unicellular based on the number of cells that they are having in their body. Apart from that, the cells are sometimes able to change their shape. Some cells are like that they can change their shape. For example, amoeba. Amoeba is a unicellular organism. It just has a single cell and it can move about, it can change the shape of its cell, and that cell is able to change its shape. But there are certain other cells which are not able to change their shape, which have a fixed shape, like the nerve cell in our body. The nerve cell or neuron that we have in our body, they are not able to change their shape. They have a fixed shape. So, a cell could be like having the capability of changing its shape or not having the capability of changing its shape. Okay. We'll move on further. We'll just start with the study of plasma membrane. But before that, if you want, you can take a screenshot so that you will be having everything at one place. Every cell has an outer layer around it, and this layer is known as plasma membrane. What is this layer called? Plasma membrane. Inside the plasma membrane, inside the cell, you will find that there is a nucleus. We'll talk about it. Then there is cell cytoplasm. And in the cytoplasm, we will see that there are many cell organelles. Various cell organelles are there that we are going to study. But first, right now, we'll be studying just about the plasma membrane. So, by this diagram, by this study, you can estimate that everything that has to come inside the cell or whatever has to pass out of the cell, that is going to pass through the plasma membrane. So, anything that will be coming inside the cell or exit from the cell, that will be passing through the plasma membrane. This plasma membrane is made up of proteins and lipids. This is very important. Remember that it's made up of proteins and lipids. Later on, I'll tell you that from where we are getting these proteins and lipids. But remember right now that this plasma membrane is made up of proteins and lipids that form this plasma membrane. This plasma membrane is semi-permeable. Semi-permeable means it will allow some things to pass through it and it won't allow some other things to pass through it. Okay. Now coming to a very important topic, that is diffusion. What is diffusion? Movement of solutes from a region of higher concentration to a region where its concentration is low, that is known as diffusion. So whenever any solutes is going from a region where it's in high amount to a region where it's low, that is known as diffusion. By the process of diffusion only, oxygen and carbon dioxide, apart from that, nutrients and waste material move in and out of a cell. I'll show you how. So, a cell requires oxygen, and during the process of respiration, every cell produces carbon dioxide. Now, every cell is surrounded by blood capillaries. These are blood capillaries which are having the oxygenated blood in them. So, oxygen is there in this blood capillaries. this oxygen moves into the cells through the plasma membrane. Because oxygen concentration is high in blood capillaries, so it will go inside the cell where it is less oxygen is there, where there is less concentration of oxygen. Apart from that, carbon dioxide from the cell comes into the blood capillaries, because these blood capillaries are having oxygenated blood. So all the carbon dioxide is removed from that. So what will happen? The carbon dioxide concentration is more in the cell. So the carbon dioxide will move from its region of higher concentration to the capillaries where there is less concentration of carbon dioxide. Similarly, nutrients like glucose, amino acids will go inside the cell from the blood capillaries, and the waste material, whatever waste is produced inside the cell, that is the nitrogenous waste, that is urea, whatever nitrogenous waste is produced by the cell, that will be going into the blood capillaries. So this movement of material, this movement of solutes across the plasma membrane from a region of higher concentration to a region where the concentration of these solutes is less, that is occurring by the process of diffusion. This process is known as diffusion. Similarly, water also moves from a region of higher concentration to a region of lower concentration. Means water moves from the area where water is in higher proportion to a region where water proportion is low. Means where water is more, from there it is going to move to the area where water is less. जहाँ पे ज़्यादा पानी है वहाँ से वो इस एरिया में मूव होगा जहाँ पर पानी कम है. And that process of movement of water across the plasma membrane is known as osmosis. So when the solutes or the gases are moving across the plasma membrane, that process will be known as diffusion. But when this diffusion process is followed by water, means the water is moving in and out of the plasma membrane, we give it a special term that is known as osmosis. That is movement of water from the area of higher concentration, higher water concentration, to an area of lower water concentration. That is known as osmosis. Okay. Now, because of osmosis only, we see many phenomena that occur, and there are different conditions that will explain us osmosis. So let's see to those conditions now. First condition, we'll see all these conditions with some daily life examples that will make your learning to be easier. So what happens when you sprinkle some salt on the cucumber? You find that after some time, there is some water in the plate that is having the cucumber slices. Okay, you slice some cucumber, put some water over, put some salt over it, sprinkle some salt over it, and after some time you will observe that there is water in the plate. From where that water has came? You just added the salt. From where that water came? So there was higher water concentration inside the cell, and now this water has moved from the cell to outside where it was solutes were there and no water was less. Okay, water was less or very less or no water was there. So from a region of higher water concentration, the water has moved to a region of lower water concentration by the process of osmosis. And when the water is moving out of the plasma membrane, that process is known as exosmosis. Now always remember, wherever there is exo word, means something is coming out. Always remember that. If you learn the meaning of these terms, that will make your learning very easy. In 10th standard, you will learn about exothermic reactions, where heat will be coming out. So remember that wherever exo word is there, means something is coming out. So here exosmosis means water is coming from the cell to outside. Water is moving outside the cell. That process is known as exosmosis. Why this water is moving out of the cell? Because there was less water outside. and there was more water inside. Means we have placed our cell in a hypertonic solution. What is such a solution known as? That solution is known as a hypertonic solution. Means a solution that is having less water but more solute is there. So such a solution is known as hypertonic solution, and whenever we place our cell in a hypertonic solution, there is exit of water from the cell. Means the water is going to come outside the cell, and that process is known as exosmosis. Now, when the water comes out of the cell, what will happen to the cell? The cell constituents are going to shrink. So, whatever the contents are there in the cell, the organelles still will be there, but that cytoplasm will be having now less amount of water in it. And so, our cell is going to shrink in this condition. What will happen to our cell? The cell is going to shrink in this situation. Okay. The reverse also happens sometimes. I'll tell you an example. That is, whenever we place we put some raisins in kheer. Okay. So, whenever you put some raisins, that is kishmish, in kheer or in water, you find that the raisins swell up. They just gain the water, they just get that water inside their cells and they are going to swell up. So, such a movement of water into the cells, when the water is moving into the cells, it's known as endosmosis. Here also, osmosis. Here also remember that endo word means something is moving inside. Endo, andar, ऐसे याद कर लेना. So, when water is moving inside the cells, that is known as endoosmosis. Okay. When this is going to happen? This is going to happen when we place our cell in a hypotonic solution. What is a hypotonic solution? A solution that is having more water in it. More water is there and less solutes are there. Such a solution is known as a hypotonic solution. So, whenever we place a cell in a hypotonic solution, then what will happen? That the water from the solution, because the solution was having more water. So, the water will be moving from the area where there was more water to the area where there was less water. And you know that raisins are the dried grapes. So, they are not having enough water. They are having very little water. So, water from outside environment is going to move inside the cells, and that process is known as endosmosis. Now, what will happen to a cell when there is endosmosis? The cell is going to swell up. Okay. So, a cell is going to swell up because of that water coming inside the cell. Okay. Now, I would like to tell you one extra thing, that if this is an animal cell, then if we just keep it on adding some water, then that cell can even burst because of water. It's just like a water balloon. So, if you keep on adding water, if you just keep on increasing the water, that what will happen? Balloon is going to swell up, but a limit is there. If you keep on adding water to that, time will come that the balloon is going to burst. Similarly, an animal cell also bursts if there is excess of endosmosis. But in case of plant cell, the cell is not going to burst. Resins come from plants, so they are not going to burst. The cells are not going to burst in case of plant cell, because in plant cell, the plasma membrane is surrounded by another layer, and that is known as cell wall. We'll learn about the cell wall in the next class. So, right now just remember that a plant cell is not going to burst even if it's placed in a hypotonic solution, because plasma membrane is surrounded by a cell wall. Okay. Now, we have seen about exosmosis, the water coming out of the cell. We have seen about endosmosis, water entering into the cell. Third type of solution could be isotonic solution. Means if the concentration of water is same outside the cell as well as inside the cell, if it is exactly the same, then what is going to happen? Then water is going to come out of the cell and the same amount of water is going to enter into the cell. Okay, the water will be continuously moving in and out of the cell, but net effect on the cell will be zero. Means when the cell is placed in an isotonic solution, Isotonic is a solution that is having the same concentration as that of the cell. So in case a cell is placed in an isotonic solution, then there will be movement of water in and out of the cell, but whatever is the amount of water entering into the cell, same amount of water will be moving out of the cell, and so the net effect of that will be zero. That will be cancelled out. The cell will neither shrink nor it is going to swell up. The cell size is going to remain the same. The cell size remains the same in case of isotonic solution. Okay, so that is about osmosis. The plasma membrane is even flexible. This plasma membrane in some organisms, in some of the cells, is really very flexible. One such example of that is the amoeba. In amoeba, the food is taken through this plasma membrane only. The food is engulfed through this plasma membrane only, and that shows that this plasma membrane is flexible. And this process of taking the food through the plasma membrane in case of amoeba, it's known as endocytosis. That is known as endocytosis. Very easy to remember. Endo means something is going inside the cell. Endo means under. And cytos word means cell. So something is going inside the cell. Okay. So the food is moving into the cell in case of amoeba through the plasma membrane, and that process is known as endoosmosis. I told you that in plants, the absorption of water by the roots is also by the process of osmosis only. Okay, so by the process of osmosis only, the roots are able to absorb the water from the soil, and that we will be learning later in another chapter. But right now you should remember up to this. Cell wall is an additional covering that is present only in a plant cell. The cell wall is absent in an animal cell. So if we examine the cell of ours or of any of the birds or any of the animals, that won't be there. But fungi, bacteria and plants have an additional covering over the cell that is known as the cell wall. So we have seen that there was a plasma membrane, and inside the plasma membrane, whatever the contents are there, the fluid part of that was known as cytoplasm. Inside the cell, there was a cell nucleus, and we have seen that this layer was known as the cell membrane. In case of plants, there is an additional covering around the cell membrane, and that is known as a cell wall. Okay. Now, this cell wall is not a continuous structure as I have drawn here. Okay. It's a double layer structure, and that is impermeable. That is made up of cellulose, and it won't allow anything to pass through it. So, how the things will come into the cell and move out of the cell? That will be moving through the pores that are present between the cell wall. So, cell wall is not continuous layer. It is having pores in between, and from these pores only anything can come across the cell. So we have seen in the plasma membrane that plasma membrane was a semi-permeable membrane. It will allow some things to pass through it, and it will not allow certain other things to pass through it. But this cell wall is completely impermeable. So all the things can pass only from the gaps that are present between the cell wall. So I'm just completing my cell wall. The cell wall is a incontinuous structure, means it is interrupted. It is having gaps in between. That are the pores are present in the cell wall, and through these pores only anything can come across into the plant cell. So this layer is cell wall. This cell wall is made up of cellulose. So cellulose is present only in plants, you heard that. So in case of plants, this cell wall is made up of cellulose. In bacteria and in fungi, the cell wall is made up of some different thing. But in case of plant cell, the cell wall is made up of cellulose. Okay. Now, it provides strength to a plant cell. In the previous video, we have seen about osmosis, and we have seen about endoosmosis and exosmosis. So let's learn once again about those processes and see how the cell wall is going to help a plant. So in case there is exosmosis, exosmosis means water moves out of the cell. then what is going to happen in a plant cell? So if the contents of the cell are moving out, if the cell is put in a hypertonic solution, then the water from the cell will move out. In case of plant cell, what is going to happen is that all the contents of the cell will be shrink and they will get collected at one place, that will be separated from the cell wall, and all the contents of the cell will get shrink and they will come to one part of the cell. Okay, and this condition, that is that aggregation of all the cell contents to one place, this is known as plasmolysis. That's known as plasmolysis. Means all the protoplasm of the cell that will be getting collected at one place, and that is known as a plasmolysis. Okay, this plasmolysis will occur only in case of we are putting the cell in a hypertonic solution, and due to hypertonic solution, the water moves out of the cell, all the cell components, this get collected at one corner, and that is known as plasmolysis. Okay, you can somewhat compare it with a washing machine. So once the clothes are spun in a washing machine, then all the water is removed from the clothes, and then you see that all the clothes are just getting settled at one corner of the washing machine. They're not all spread in the washing machine, they're just getting collected at one corner, and similarly in case of plant cell also. Okay, next, what would happen when endosmosis is there? I told you in the previous video also that in case there is endosmosis, means a plant cell is kept in a hypotonic solution. When the plant cell is kept in a hypotonic solution, then water from outside will enter inside the cell and the cell is going to swell up. If it is an animal cell and too much of water enters into the cell, then the cell can burst. So the cell cannot withstand a very like a hypotonic solution. If it's an animal cell, it won't be able to withstand that hypotonic solution. Won't be able to withstand means usko jhel nahi paega. So it is going to burst. It will not be able to survive with that hypotonic solution. But in case of a plant cell, there will be no effect on the cell. Even if a lot of the cell is kept in a hypotonic solution and water enters into the cell, this cell wall is going to protect the cell from being burst. So the cell is not going to burst. The cell can withstand that hypotonic solution. Even after the entry of water, the cell wall is there around it, and so it will protect the cell from being burst. Okay, so that is about the cell wall. It's an additional covering that is present in case of plants, fungi, and some bacteria have this cell wall. Okay, now coming to another component of the cell, that is nucleus. Okay, so nucleus is the main regulatory structure that is present in a cell. And I hope you remember who discovered the nucleus. Think about it. We just learned that in the first video. That was Robert Brown. So Robert Brown was the scientist who discovered the nucleus. He first observed the nucleus. So let's see what is the nucleus and what is its function and what's the structure of nucleus. So in the cell, there is a nucleus present, and in case of plant cell, this nucleus is at one corner, and in case of animal cell, this nucleus is at the center of the cell. Okay, we'll learn about it in the next video that why in plant cell it's at one corner, but right now just learn about the structure of the nucleus. So nucleus is a double membrane structure. It is having double membrane, and this membrane is having some pores. So like we have drawn the pores in the cell wall, similarly the nuclear membrane is also having the pores. This is a nuclear membrane. You can see that the nuclear membrane is having some pores, some gaps are there between the nuclear membrane, and through these gaps only, these gaps are known as nuclear pores. Nuclear pores. And through these gaps only, whatever things have to be getting transferred from the cytoplasm into the nucleus or from the nucleus to the cytoplasm. So whatever movement of substances is there, that is going to happen through these nuclear pores only. So exchange of material between the cytoplasm and the nucleus will be taking place through these nuclear pores. Okay. Apart from that, inside the nucleus, there are rod-like structures, and that are known as chromosomes. So some rod-like structures are visible inside the nucleus, and these rod-like structures are known as nucleolus, that are known as chromosomes. And apart from chromosomes, there is a nucleolus in the nucleus. Okay, so nucleolus is the aggregation of proteins that is present in the nucleus. Apart from that, chromosomes are there. Now, what is chromosomes? These chromosomes contains the genetic information, means the color of your eyes, your blood group, the texture of your hair, and many things that are decided by the genes. That all information is stored in these chromosomes. So these chromosomes are present in the nucleus of a cell. Now, these chromosomes are made up of DNA and proteins. DNA and proteins make up this chromosomes. And what is DNA? DNA means deoxyribose nucleic acid. This DNA only contains the genetic information. Means DNA is like in 11th standard you will read about the structure of DNA. So I'm just telling you very basic about it. And it has a double helical structure. Double helical structure is somewhat like that spiral stairs that you have seen. Okay, sometimes the stairs are spiral, not circular like that. So such a structure is there of DNA, and on the DNA there are various segments that are known as genes. Okay, there are various segments that are known as genes. So various segments are there, small segments are there, and that are known as genes. And each gene contains a specific information. For example, one of the genes will be deciding the color of your eyes. Another gene could be deciding your blood group. So these genes are present on the DNA, and the DNA is there. DNA along with the proteins forms the chromosomes. Okay, it's a very common thing for the students to get confused about what are genes, what is DNA, what are chromosomes. Okay, so it's all like similar only because genes are present in the DNA, and DNA along with proteins forms the chromosomes. These chromosomes, as I told you, are rod-like structures. They are visible as rod-like structures. But these will be appearing as rod-like structures only when the cell is just about to divide. When the cell is about to divide, the chromosomes will be visible as rod-like structures. But if the cell is right now not going to divide, if the cell is newly formed, it's just growing, then these chromosomes are not visible inside the nucleus. What is visible inside the nucleus is thread-like structures. These are known as chromatin threads. So some thread-like structures are visible inside the cell, and that are known as chromatin threads. These chromatin threads only come close to each other, aggregate, and forms the chromosomes when the cell is just about to divide. Okay, so it's very important that these chromosomes will be visible only when the cell is just about to divide. So if you are able to see a cell, and if you are observing a cell, and if you are able to see the chromosomes, that shows that the cell is going to divide. Now, what is the importance of nucleus in a cell? Nucleus is a very important structure. Nucleus plays a major role in regulating all the functioning of a cell. So it's the regulating body, like the principal in our school, that is the regulating body for the cell. So everything will be regulated by the nucleus. Apart from that, cellular reproduction. Cellular reproduction means one cell will be divided into two cells. So the division of the cells will also be controlled by the nucleus. First, the nucleus divides. Whenever cell has to divide, first the nucleus divides, and then the cytoplasm also divides, and one cell gets converted into two cells. Okay, so for cellular reproduction, apart from that, for regulating all the functioning of the cell, this nucleus is very important. But do you know there are certain organisms which do not have a membrane around their nucleus? The nuclear material, the nucleic acid, like deoxyribose nucleic acid, is there inside that inside the nucleus, I told you. So in some of the organisms, this nucleic acid is just present without any covering around it, and that is known as a nucleoid. Okay? So such organisms which do not have a nuclear membrane are known as prokaryotic organisms. Some bacteria are there which are prokaryotic organisms. Prokaryotic means, pro means primitive, and karyot means cell. So primitive cell is there. Primitive, very basic cellular structure is there. So in some of the organisms, in some of the bacteria, it is seen that this is a cell. then inside the cell, the nucleic acid is present just like that. It's not surrounded by a nuclear membrane. Such organisms which do not have a nuclear membrane around their nucleus are known as prokaryotic organisms. So what do we call the organisms which have a nuclear membrane? Such organisms like human beings are known as eukaryotic organisms, means we have a nuclear membrane around the nucleus. Okay. So instead of nucleus, their nucleic acid is present, and such is known as nucleoid. Such a structure, that is aggregation of nucleic acid inside the cell, that is not called a nucleus. Instead of nucleus, we call it as nucleoid. Okay. Now, next very important question, very important question for exams is difference between prokaryotic and eukaryotic organisms. So let's work it out. Prokaryotic and eukaryotic. The first point is about the size. The prokaryotic cell is having a very small size, usually from 1 to 10 micrometer. The size of a prokaryotic cell is usually small, and it's approximately somewhere between 1 micrometer to 10 micrometer. What is micrometer? 1 micrometer means 10 to the power minus 6 meters. Wherever micro word is there, that refers to 10 to the power 6. Okay. And in case of eukaryotic organism, the size of the cell is approximately 5 micrometer to 100 micrometer. Okay, that is the size of the cell. Now, the basic difference between prokaryotic and eukaryotic is nuclear membrane is absent. So when you are writing the differences, just write the points and then write the differences. So the first point will be size. The second will be nuclear membrane, and in this nuclear membrane in prokaryotic you will write that nuclear membrane is absent, and in case of eukaryotic organisms the nuclear membrane is present. Apart from that, the nuclear the nuclear material in case of prokaryotic organisms is ill-defined, and that is called as nucleoid. The nucleic acid that is present, it's ill-defined, that is not having a perfect boundary around it, and that is called as nucleoid. But in case of eukaryotic organism, the nuclear material is well defined and it is present in the confined area only. It is there inside the nuclear membrane. So nuclear membrane is present and the nuclear material is present inside this nuclear membrane only. Okay. Now, if we talk about the number of chromosomes in prokaryotic organism is just 1. So the third point will be number of chromosomes. And in case of prokaryotic organism, how many chromosomes are there? 1 chromosome is there. And in case of eukaryotic organism, many chromosomes are there. For example, in human beings, there are 46 chromosomes. Okay, 23 pairs of chromosomes are there in human beings. So it depends from species to species, but the number of chromosomes in prokaryotic is just 1, and the number of chromosomes in eukaryotic organism is more than 1. Okay. Apart from that, the prokaryotic organisms do not have membrane-bound cell organelles. So if we just observe the cell organelles inside the cell, we will see that inside the cytoplasm there are various cell organelles like mitochondria is there. In case of plant cell, chloroplast is there. So all these are having a membrane around them. But in case of prokaryotic organism, membrane-bound cell organelles are not there. The organelles are not having any membrane around them. Okay, nucleus is also not having any nuclear membrane. The cell organelles are also not having any nuclear and are not having any membrane around them. So, organelles, the next point will be organelles. Organelles in case of prokaryotic organism do not have any membrane around them, and in case of eukaryotic organisms, they have membrane-bound cell organelles. Okay. So that is the difference between prokaryotic and eukaryotic organisms. That's a very frequently asked question in your exams for 2 marks. Okay. Now comes about the importance of membrane around any of the structures. Membranes are very important. For example, plasma membrane or cell membrane that we have discussed. One of the organisms like viruses do not have a membrane around them, and so they are not strictly defined as living organisms even. They are like just at the junction between living and non-living organisms. Why I am saying so? Because outside a host body means when the virus is not inside any of the organisms, it's not there inside any animal, it's not there inside any plant, then it just behave like a dead cell and it's not able to carry out its functioning. But when it enters any of the organism, it uses the genetic machinery of that organism, and then it starts its reproduction, then it starts its growth, multiplication, everything is there. So viruses use the machinery, cellular machinery of the organism in which they are entering, and they do not have a cell membrane around them, and they are not strictly known as living cells even, because of the absence of the capability to divide on their own. They can't divide outside the body of a host. Okay, isn't that interesting to know about viruses? Okay, so that was all about nucleus. Then we come to cell cytoplasm. Whatever components are there inside the plasma membrane, but outside the nucleus, that is all the fluid part is there, and that fluid part is known as cell cytoplasm. In this cytoplasm only, different cell organelles are there. What are organelles? Organelles are small structures about which we are going to study in detail in the next video, that many cell organelles are there inside the cells. So all these organelles are present inside the cell cytoplasm only. Okay, so that cytoplasm is present, and inside the cytoplasm we have different cell organelles about which we are going to study in detail one by one, considering all the organelles. In this part of the video we are going to discuss about the various cell organelles. What are cell organelles? Human body has many organs that combine together and helps us to perform different functions. Similarly, in a cell also, there are various small parts, small components of a cell, and they together help in the functioning of a cell. A cell is known as a basic structural and functional unit of an organism. Why is it called so? A cell is known as a structural unit because like bricks helps us to make a house. Similarly, cells together combine and form the whole organism. So a basic structural unit of an organism is a cell. Why is it called a functional unit? Every cell is capable of performing cellular respiration, that is breakdown of glucose to produce energy. Apart from that, every cell can perform excretion, that is removal of waste from it. Every cell performs digestion and nutrition, that is takes up the glucose, amino acids and use it for preparing the different cell components. And that is why every cell can be said as a functional unit, because every function that is performed by an organism as a whole, that could be performed by each and every cell as well. That is why we call a cell as a structural and functional unit. So now let's look about the functions and the structure of various cell organelles. Okay, starting with the first organelle, that is endoplasmic reticulum. Endoplasmic reticulum, here you can see that it's present around the nucleus, and it's a form of tubules. Long tubules are there. Long tubules as well as vesicles forms the endoplasmic reticulum. This endoplasmic reticulum is very important in a cell because it helps in transportation. Endoplasmic reticulum will be helping in transportation of material from nucleus to cytoplasm, from one part of the cytoplasm to another part of the cytoplasm when the things have to be transported. Apart from that, when the things have to be transferred from the nucleus up to the plasma membrane, then also this endoplasmic reticulum is going to help in the transport of material. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough word itself suggests that its surface is not going to be very smooth. But why is it rough? Rough endoplasmic reticulum is rough because of presence of small granular structures called ribosomes. Ribosomes are present over the surface of endoplasmic reticulum, which makes its surface rough. It's just like putting some sand over the road. Will that make the road rough? Similarly, these small granular structures are there that are called ribosomes, about which we are going to discuss more, and that makes the surface of this endoplasmic reticulum to be rough. And that is why a reticulum, endoplasmic reticulum that is containing these ribosomes is known as rough endoplasmic reticulum. If the endoplasmic reticulum doesn't has any ribosomes over its surface, then it's called as smooth endoplasmic reticulum, represented by SER, smooth endoplasmic reticulum. Okay, so endoplasmic reticulum we have seen it's of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, this rough endoplasmic reticulum, I told you, it contains ribosomes, and ribosomes helps in the synthesis of proteins. Ribosomes are also known as the protein factory of a cell. So these ribosomes helps in the synthesis of proteins. And do you all know enzymes, hormones, as well as all the cell membranes, they all contain proteins. Apart from that, this rough endoplasmic reticulum is having the ribosomes, that is manufacturing proteins, and hence the one of the function of rough endoplasmic reticulum will be synthesis of proteins. What's the function of smooth endoplasmic reticulum then? Smooth endoplasmic reticulum helps in the synthesis of lipids. Lipids are also known as fats. So, lipids will be synthesized by smooth endoplasmic reticulum and proteins are synthesized by rough endoplasmic reticulum. Do you all remember while teaching plasma membrane, I told you all that plasma membrane is made up of proteins and lipids, and I asked you all to remember that. From where these proteins and lipids are coming? I hope now you are able to guess that. That is from the endoplasmic reticulum. So, proteins required for plasma membrane synthesis comes from rough endoplasmic reticulum, and the lipids required comes from smooth endoplasmic reticulum. And that is why this endoplasmic reticulum, one of the functions is membrane biogenesis. It's very important as an individual question also, that what is membrane biogenesis? Formation of plasma membrane requires proteins that are synthesized by rough endoplasmic reticulum and lipids that are synthesized by smooth endoplasmic reticulum. And that is why endoplasmic reticulum helps in the formation of plasma membrane, which is known as membrane biogenesis. In liver cells, the smooth endoplasmic reticulum also helps in the detoxification of drugs, medicines, poisonous substances. So whatever medicines we take, that also will be detoxified by the liver by the help of smooth endoplasmic reticulum present in the liver cells. I hope that is clear. Apart from that, I told you that enzymes, hormones, they are also made up of proteins and lipids only. So these proteins and lipids are also used for the synthesis of enzymes and hormones in the body. We will read more about enzymes and hormones later on. Next is endoplasmic reticulum also provides a structural framework of a cell. What is the meaning of structural framework? Whenever there is a construction of a building going on, you must have observed that first some iron rods are fixed and around which the construction of bricks is done. So that framework is provided by those iron rods. Similarly, in a cell also, the basic function of maintaining a proper shape and providing a proper support to a cell is done by endoplasmic reticulum. It's spread all in the cell. It is also going to connect it is going to be connected even to the plasma membrane. So it's present all over the cell, and so it forms a basic structural framework of a cell. Apart from that, if you microscopically observe the endoplasmic reticulum, its structure is similar to that of plasma membrane. So the structure of plasma membrane and endoplasmic reticulum are similar. One more thing that you can observe is endoplasmic reticulum is having a single membrane. So it's a single membrane bound structure. So I hope all this is clear to you all. You are able to understand about endoplasmic reticulum. Now I'll shift aside that you can take a screenshot if you want, and that's all about the endoplasmic reticulum. The next organelle that we are going to study about is mitochondria. Mitochondria is known as the powerhouse of the cell. So let's see to the structure of this powerhouse. Mitochondria is a double membrane bound organelle. It is having two membranes. It's a very special and a very important cell organelle. The outer membrane is porous, and the inner membrane, you can see that it has many folds. These folds increase the surface area. Why we need to increase the surface area? Because ATP synthesis is going to take place here. Why ATP synthesis? In our cells, every cell is going to perform cellular respiration. Cellular respiration means glucose will be broken down into its components, that is carbon dioxide and water, and energy will be released. This energy will be stored in the form of ATP, adenosine triphosphate. So this process of breakdown of glucose to form ATP, and this ATP storage, it's done in the mitochondria. ATP is also known as the energy currency of a cell. Remember, it's very important. What is known as the energy currency of a cell? So that is ATP molecules, adenosine triphosphate. ATP is known as the energy currency of a cell. So this ATP synthesis is going to take place in the mitochondria, and that is why it is known as the powerhouse of the cell, because ATP is the energy currency, and whenever you are storing some energy, the place where you are storing the energy, that is called powerhouse. So mitochondria is known as the powerhouse of a cell. It's a very special organelle in the sense that it has its own DNA and own ribosomes. So it can divide on its own, it can synthesize proteins on its own. So very few organelles are there that that have their own DNA and ribosomes, and that is mitochondria. The other one we'll read in plant cell, that is chloroplast, that is also having its own DNA and ribosomes. All that much is given in your NCERT book, but I would like to tell you something extra from this topic. Actually, cell topic, especially the cell organelles, are very important for you all. In NEET also, you will be having some questions based on this topic. So it's really very important. And one more thing is there, you will be reading about cell once again in 11th standard as well. So it will be really better for you all that if you're preparing for NSO exams, or if you are preparing for some competitive exams later on, this will be useful for you to gain some extra knowledge. So here what I have written is that's given in your NCERT book. And now I'll be telling you something extra, not given in NCERT book, still it's very important. Okay. So, who discovered the mitochondria? Mitochondria was first of all discovered by Kolliker. Kolliker is a scientist who first saw the mitochondria, observed the mitochondria for the first time. But the name mitochondria was given by another scientist known as Benda. So Benda was the first scientist to give the name mitochondria. Mitochondria is a plural and mitochondrion is a singular term used for a single mitochondria. I told you that the outer layer is porous and it's continuous. The inner layer is folded, and these folds are known as cristae. And whatever matter is there inside this inner membrane, the inner portion, whatever is here, that is called as matrix. On the cristae, there are, you can see, some bead-like structures are there, and they are known as oxysomes. These oxysomes contains the enzymes that are required for ATP synthesis. So we have seen in NCERT also it's given that ATP synthesis is going to occur here. But how that is possible? Because of oxysomes which are present on the inner lining of the cristae, and that is having the enzymes for synthesis of ATP. That is why mitochondria is a site for cellular respiration. Okay, cellular respiration means every cell is performing respiration, that is breakdown of glucose to release carbon dioxide and water. Okay, this breakdown of glucose requires oxygen, so breakdown of glucose in presence of oxygen to release carbon dioxide and water, that is known as cellular respiration. Okay, so that's all for mitochondria. I'll shift to side so that you can take a screenshot. Our next cell organelle is Golgi apparatus. Golgi apparatus was discovered by the scientist Camillo Golgi. He was a great scientist. He even got a Nobel Prize for his work that he did for the staining of nerve cells. And the reaction that he carried out is known as black reaction, that helps to study nervous system by staining a neuron. So he got even a Nobel Prize for that. So it was a great scientist, Camillo Golgi, who discovered the Golgi apparatus. Now, what's Golgi apparatus and what's its function? So, the structure first. Golgi apparatus is made up of a single membrane bound vesicles which are arranged almost parallel to each other in a stack. Now, what is a stack? Stack means one above the other. So, if you put a stack of coins, means putting the coins one over the other. That is called a stack of coins. So, the Golgi apparatus is made up of single membrane bound vesicles which are arranged almost parallel to each other, and it's like a stack, and that is known as cisterns. This stack of Golgi apparatus, this stack of vesicles is known as cisterns. So that is the structure of Golgi apparatus. This Golgi apparatus is in close connection with endoplasmic reticulum. It's present close to the endoplasmic reticulum. That is also for a reason. Endoplasmic reticulum, you all know that it synthesizes proteins and fats. So, rough endoplasmic reticulum synthesizes proteins, smooth endoplasmic reticulum synthesizes fats. So, endoplasmic reticulum basically synthesizes proteins and fats. And the function of Golgi apparatus is the storage, modification, and packaging of all these products in vesicles and sending them to the places where they are required. We can understand the functioning of Golgi apparatus comparing it with a chips factory. So suppose our endoplasmic reticulum is just like a farm. Any factory, if you have to set up, that you prefer to set up to a place which is close to the area where there are raw materials required for that. So if we talk about a chips factory, then the raw material is potato. So suppose the potatoes which are grown in this endoplasmic reticulum, and the chips factory, the Golgi apparatus, is going to store the potatoes. It's going to modify it into the form of chips and package that and send it to you. So similarly, the Golgi apparatus is close to the endoplasmic reticulum, that is close to the raw materials which are needed to be stored, modified, and packaged and sent to the areas of action. Golgi apparatus also performs the functioning of converting simple sugars to complex sugars. So that also, that modification of simple sugars into complex sugars, that will also be performed by Golgi apparatus. Apart from that, one very important function of Golgi apparatus is the formation of lysosomes. Lysosomes, we are going to talk about that. but lysosomes are formed by the Golgi apparatus, because lysosomes also has some enzymes, and enzymes are made up of proteins only. So this Golgi apparatus will be helping in the formation of lysosomes and plasma membrane, which again is made up of proteins and lipids. So formation of lysosomes and plasma membranes will also be helped by the Golgi apparatus. I hope this is an easy organelle for you all to remember. I'll just shift aside so that you can take a screenshot. The cell organelle that we are going to discuss now is lysosomes. Lysosomes are a single membrane bound vesicle structure, and it is containing digestive enzymes that are prepared by rough endoplasmic reticulum. I told you earlier also that rough endoplasmic reticulum prepares proteins, and enzymes are a form of proteins only. So the digestive enzyme that is present inside the lysosomes, that is prepared by rough endoplasmic reticulum. Apart from that, remember that lysosomes are formed by the Golgi apparatus. Now, the lysosomes has a very important function in a cell, that forms the waste disposal system of a cell. Why it's said so? The lysosomes contain the digestive enzyme. So whenever any bacteria or some cell organelle is worn out, for example, if a mitochondria is worn out, it's damaged, so it has to be removed from the cell. So all such damaged cell organelles, any foreign body like bacteria entering inside the cell, that all will be removed by the lysosome. So it's known as a waste disposal system of a cell. And it's also known as the suicidal bags of the cell. Suicide means killing oneself, and lysosomes are known as suicidal bags because if a cell is old, damaged, or worn out due to some reason, then these lysosomes are going to burst. And when it bursts, this digestive enzyme is going to spread into the entire cell and digest all the cell components. So the cell in which it was present, it is digesting that particular cell only, and so it's known as a suicidal bags of a cell. And it also helps to keep the cell clean by removing any waste, any of the dead material from the cell, and helps the cell to be kept clean. That is another function of lysosomes. That's all what we discussed right now. It's given in your NCERT book, but I would like to add something extra, not given in NCERT, but again important for your 11th standard biology, important for your NEET exams. So that is discovery. The lysosomes were discovered by Christian D. Dew. D. Dew was a scientist who discovered the lysosomes. And I told you it is having a digestive enzyme. What's the name of the digestive enzyme? That is hydrolytic enzyme. The digestive enzyme present in lysosomes is hydrolytic enzyme. And these are also known as scavengers in a cell. Why called scavengers? Scavengers means organisms which which eat up the dead and decaying matter. So they are also removing the dead cells, dead organelles, and so it's known as a scavengers in a cell. That's all about lysosomes, an important topic for your exams. So I'll just shift aside. You can take a screenshot. The next cell organelle that we are going to discuss now is present only in a plant cell and absent in animals, that is plastids. Plastids could be having some color, then they are known as chromoplast. Chroma word means color. So chromoplasts are the plastids that have any color in them, and if they don't have any color, that is they are colorless or white in color, then they are known as leucoplast. Wherever the word leuco comes, that refers to white color. So leucoplasts are the colorless plastids, and the colored plastids are known as chromoplast. This chromoplast could be having different colors like yellow, orange. You must have seen the petals of the flowers having beautiful different colors, or you see different vegetables having different colors like brinjal. Whenever you see some fruits having different colors like red color of apple or orange color of orange, then that is because of the presence of chromoplast. But If the chromoplast is having green color, then it is giving a special name, and that name is chloroplast. Chloroplast are the plastids that have the green pigment in them, and that is the chlorophyll containing plastids. This chlorophyll containing plastids are definitely going to help in the process of photosynthesis performed by the plants. That is why the chloroplasts are also known as the kitchen of the cell, because in the kitchen the food is prepared. Similarly, in the leaves the food is prepared in the chloroplast cells, chloroplast organelles. So they are known as the kitchen of a cell. So name the cell organelle that is known as the kitchen of a cell, that is chloroplast. Now, if it's white in color, that is leucoplast, that is the colorless plastids. They are usually present in the roots and in the stem. In the roots sometimes, or in the stem, there is storage part of the plants, like potatoes. Potatoes are a modified stem only. Apart from that, sweet potato, that is a modified root. So they are the storage part of the plants where the food is being stored by the plants, and they contain the leucoplast. So leucoplast has performed the function of storing of starch. It could be oils, it could be proteins. So proteins, oil, starch could all be stored in the leucoplast. Plastids are a special organelles that have their own DNA and own ribosomes. Do you remember one more cell organelle that I told you it's having its own DNA and ribosomes? Think about it, recall that. Yes, that is mitochondria. So mitochondria and plastids are the only cell organelles that they have their own DNA and ribosomes. Apart from that, plastids, that you need to remember is that they are a double membrane bound structure. They are having two membranes around them. So it's a double membrane bound structure. Now, talking about something extra that's not given in NCERT book. First is about the term plastids was given by Ernest Haeckel. E. Haeckel, you can remember, or Ernest Haeckel gave the term plastids. Apart from that, the structure of plastids is really important and interesting also. I told you it's a double membrane bound structure, so you can see the outer membrane and the inner membrane, both are continuous. Apart from that, inside the plastids, there is like a structure like a stack of coins. Stack of coins means coins placed one above the other. So that single coin is known as thylakoid. That single coin like structure is known as thylakoid. And these coins placed one below the other, that complete stack of coin is known as granum. Why is it important? Because in photosynthesis, you will read in higher classes, there are two reactions involved. One is light reaction and the other is dark reaction. So in NSO exams or in competitive exams, you are asked that where the light reaction of photosynthesis occurs. So the light reaction of photosynthesis occurs in the granum part of chloroplast. Now, where does this dark reaction occurs? So, dark reaction occurs in the matrix. Matrix is the filling. Whatever is there inside that space is there inside the chloroplast, that is the matrix. So, this matrix is having stroma. That matrix part is known as stroma. And that stroma is a site where there is dark reaction of photosynthesis. So, light reaction occurs in the granum part and dark reaction occurs in the stroma. And this is the organelle I told you earlier also, that it is having its own DNA and its own ribosomes also. Okay, so that stack of coins is there. A single coin is known as thylakoid. That complete stack of coins is known as granum, where the light reaction of photosynthesis is going to take place. The other structure is the matrix, that is the stroma, where the dark reaction of photosynthesis is going to take place. Okay, so that's all about the plastids. What you really need to remember is it's a double membrane bound structure having its own DNA and own ribosomes. It could be colorless, that is leucoplast, or colored, that is chromoplast. If it's colored and the color is green in color, that specially is known as chloroplast. Okay, the cell organelle which is known as the kitchen of the cell, that is also chloroplast. So I'll just shift aside. You can take a screenshot. Our next cell organelle is vacuole. Vacuole is present in a plant cell as well as in an animal cell. But in animal cell, it's very small, and in plant cell, it's really big. The plant cell have a big vacuole which stores amino acids, proteins, sugars, organic acids like citric acid in lemon and oranges. So these are known as organic acids that are all stored in a vacuole. The fluid present in the vacuole is known as cell sap. In case of plant cell, the nucleus is pushed to one corner of the cell because of the presence of a big vacuole. Whereas in case of animal cell, the nucleus is at the center of the cell because the vacuole is very small. The vacuole is also used by the plants to remove the waste from the plant cells. For example, the plant has to remove some waste, so it can store it in the vacuole of the leaves, and now when the leaves will be shed off, along with the leaves, the vacuole will be taking away the excretory products of the plants as well. So plants use the vacuole as a method of excreting out their waste products as well. In case of amoeba, the food is also stored in a vacuole. So, amoeba takes in the food through its cell membrane and forms a food vacuole around the food material. And then the food will be digested by amoeba. One more thing is there, that is rigidity and turgidity is provided by this vacuole. If you see a fresh tomato, you see that the skin is very tense, it's tight, and that is only known as rigidity and turgidity. That is provided by the fluid content in the vacuole. In higher classes, you will read that cell sap vacuole, food vacuole are actually the types of vacuole. But in 9th standard, these types of vacuoles are not mentioned. One extra thing that I would like to add is the membrane around the vacuole is known as tonoplast. That is known as tonoplast. So that's all about vacuole. I'll just shift aside so that you can take a screenshot. Next to cell organelles are both membraneless, that is, they do not have any membrane around them. So the first one is ribosomes. Ribosomes are known as the protein factory of a cell. They help in the synthesis of proteins. Ribosomes are seen over the rough endoplasmic reticulum, found scattered in the cytoplasm, means here and there in the cytoplasm you can see many ribosomes. Apart from that, they are also present in mitochondria and plastids. So, the ribosomes are present on rough endoplasmic reticulum, mitochondria, plastids, and scattered in the cell cytoplasm. One thing that I would like to add that's not given in NCERT is the types of ribosomes. Ribosomes are of two types. 70S ribosomes are seen in prokaryotic organisms, and eukaryotic organisms have 80S type of ribosomes. Okay, so that's not mentioned in NCERT book. Next cell organelle is centrosome, about which in the NCERT book it's only shown in the diagram. Nothing is described about the centrosome in your NCERT book. So if you want to skip about it, you can skip that. But it's important for 11th standard biology as well as NSO exams. So I would like to tell you about it. Centrosome is made up of two perpendicular structures that are cylindrical in shape, and that is known as centriole. These centriole divide from each other, separate from each other at the time of cell division, and they go to opposite parts of a cell during the cell division. They play a very important role in cell division in case of animal cell. The centrosome is present only in an animal cell and absent in plant cell. So that's all about the different cell organelles. Now we are just going to discuss about the difference between plant cell and animal cell, and we are done with our all the cell organelles. I'll just shift aside. You can take a screenshot. Now let's discuss about the difference between the plant cell and animal cell, which is a very important question for your exams. The first difference is based on the size. A plant cell is usually larger in size as compared to an animal cell. The second point is cell wall. It is present in a plant cell whereas absent in animal cell. Third point is about plastids. We have seen about chromoplast, chloroplast, leucoplast. So plastids are present only in a plant cell and absent in animal cell. Vacuole. A large vacuole is seen in a plant cell, whereas a vacuole is small in size in case of animal cell. Next is nucleus. In case of plant cell, the nucleus is usually shifted to periphery, that is at one corner of the cell because of a large vacuole. Whereas in animal cell, the nucleus is usually at the center. So I'll just shift aside. You can take a screenshot. Now at last we'll see to some things to remember for competitive exams, NSO exams and all. So first is the organelles could be divided into three categories. Some organelles are membraneless, means they do not have any membrane around them, and that are ribosomes and centrosome. Second is single membrane bound organelles. Those organelles which have a single membrane around them, and that is Golgi apparatus, lysosomes, endoplasmic reticulum, and vacuole. You can remember it by that Golu loves eggs very much. Next is double membrane bound organelles, that are nucleus, that is having a double membrane, mitochondria and plastids. One last thing that I would like to tell you is that human RBC, when it's in the mature stage, it doesn't has any nucleus, mitochondria or Golgi apparatus. So when it's getting mature, when it's developing, at that stage of development, the human RBC has these organelles, but once it matures, when it's developed fully, then the nucleus, Golgi apparatus, mitochondria, they are all not present in a mature human RBC because that place will be occupied by a big hemoglobin molecule. Okay, so that is there in mature RBC in human beings, and not only human beings, in all mammals it's that. Okay, so you can take a screenshot if you want. We have seen in detail the function and the structure of different cell organelles. Today we are going to just revise the structure of plant cell as well as animal cell and see the position of different cell organelles. I'll just shift aside so that you can take a screenshot of the diagram of plant cell and animal cell, and then we'll revise all the cell organelles, their position and their functions in a cell. So you can take a screenshot. I hope you have taken a screenshot. So let's start our revision with the animal cell. In an animal cell, the outermost layer is the plasma membrane. The plasma membrane, I told you, it's semi-permeable. It will allow the things to pass through the cell. Apart from the plasma membrane, the whatever contents are there inside the cell, that is known as protoplasm. Protoplasm includes the nucleus that is present in the cell and the liquid part of the cell, that is the cytoplasm. So protoplasm includes cytoplasm and nucleus. Now coming to the nucleus. This nucleus is there in a cell, and in case of animal cell, this nucleus is at the center position, usually present at the center of the cell. The nucleus has a membrane around it, and that is called a nuclear membrane. If an organism is not having a nuclear membrane, then the nucleus is not called nucleus, it's called nucleoid, and that is present in prokaryotic organisms, all that we have seen. So if an organism is having a nucleus, having a nuclear membrane, then it is called a eukaryotic organism. So we are seeing an animal cell, that is a eukaryotic organism. So nuclear membrane is there. Inside the nucleus, that is there is a nucleolus and the chromatin material. This chromatin material contains the DNA, and you all know that we study a lot about DNA. DNA is very useful in forensic study also, in crimes also. DNA is very important because DNA of every person is unique. So your DNA is not going to be exactly that like of your parents even. So DNA is something that is unique to every person, and that is why study of this DNA is very, very important. So that is there in a nucleus. Then from the nucleus, this nuclear membrane is there, that is double layer. Okay. So in a cell we have three organelles that have a double membrane. The first one is nucleus. Okay. Now apart from nucleus, the two other organelles that have double membrane are mitochondria, and in case of plant cell that is chloroplast. Okay. Now coming just to animal cell only. From the nucleus, the things will be transported to the cytoplasm, and from the cytoplasm, the things will be transported to the nucleus. Apart from that, from one part of the cytoplasm, the things will be transported to other part of the cytoplasm through a network of tubules, and this is known as endoplasmic reticulum. This endoplasmic reticulum is having a single membrane. Endoplasmic reticulum could be having some ribosomes over it. We have already seen that ribosomes are known as the protein factory of a cell that synthesizes proteins. Endoplasmic reticulum could be having some ribosomes over it. If the endoplasmic reticulum has ribosomes over it, then it is known as rough endoplasmic reticulum, or in short known as RER. Sometimes the endoplasmic reticulum may not be having any ribosomes over it, and then it is known as smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum, we have seen that it helps in the synthesis of lipids, and rough endoplasmic reticulum helps in the synthesis of proteins. Now, coming to another cell organelle, that is Golgi apparatus. Golgi apparatus is made up of different vesicles or cisterns that are arranged parallel to each other, and these Golgi apparatus are present close to the endoplasmic reticulum. Why it's present close to the endoplasmic reticulum? Because endoplasmic reticulum is going to synthesize different things, and Golgi apparatus is going to pack those things, store those things, and just going to modify that and send it to the places where it's required. So it has to be close to the endoplasmic reticulum so that it can perform the functions, because the functions are related to each other. Then a cell has mitochondrion. Okay, singular is mitochondrion and plural word is mitochondria. Okay, so a cell has mitochondria, and mitochondria is also a double membrane bound structure known as, mitochondria is also known as the powerhouse of a cell. You remember that, I hope. Then in a cell there are lysosomes. Lysosomes are known as the suicidal bags of the cell that are present in a animal cell. In animal cell, lysosomes are very important. In case of plant cell, they also have lysosome in them, but in case of plant cell, they are not very important. But in case of animal cell, lysosomes are very, very important, because in plant cells, usually the dead cells are not removed from the plant. The plant keeps its dead cells also. You will read in the next chapter, that is tissues, that plant has many dead cells also. But in case of animals, we don't keep our dead cells in our body, we just remove them. So that removal will be helped by lysosome. That the next organelle that is present only in animal cell and absent in a plant cell, that is centriole. Two centrioles are there that are perpendicular to each other, and they together form the centrosome. The centrosome helps in the cell division. So when an animal cell divides, these centriole will just divide from each other. They will go to opposite sides of a cell. So here we have drawn the centriole. Okay. Apart from that, ribosomes are there, that are the protein factory of a cell. These ribosomes are not having any membrane around them. So centriole as well as ribosomes, they don't have any membrane around them, and the ribosomes are scattered all through the cytoplasm. all through the cytoplasm we can see the ribosomes. Apart from that, ribosomes are visible on the rough endoplasmic reticulum as well as inside the mitochondria. Okay, so that is all about the structure of an animal cell. Whatever components are there, that is a fluid part of the cell, that is known as cytoplasm. Okay, so whatever fluid part of the cell is there, that is containing these different small structures known as organelles, that fluid part of the cell is known as cell cytoplasm. Okay, now coming to plant cell. So a plant cell also has the plasma membrane, but apart from plasma membrane there is an additional covering, outermost covering you can see, that is the cell wall. So cell wall is the outermost covering that gives a further support and strength and helps a plant cell to maintain a fixed shape. So that is present only in a plant cell. Apart from plant cell, fungi, some bacteria also have a cell wall. This cell wall in case of plants is made up of cellulose. Now, in the cell, whatever components are there inside, that the fluid part is there, that is cell cytoplasm, that is in same in plant cell as well. In plant cell also, there is a nucleus and that is containing the genetic material, that is chromatin threads and the nucleolus. But you can see in a plant cell, this nucleus is just shifted to one corner. It's not at the center, because in case of plant cell, there is a huge structure known as vacuole. This vacuole is so big that it has pushed a nucleus to one corner in case of a plant cell. So the nucleus is not present at the center, but it's at the periphery, but it's at one end of a plant cell. Then we have the Golgi apparatus in case of plant cell also, that we will be drawing close to the endoplasmic reticulum. In plant cell also, there will be both rough endoplasmic reticulum as well as smooth endoplasmic reticulum. One structure that is present only in plant cell and absent in animal cell, that is having a double membrane around it, and that will be helping in the process of photosynthesis as well, that is the presence of plastids. Okay, so we have seen about plastids, and here I have drawn chloroplast. Chloroplast is a type of plastid that we have drawn. So chloroplast is there, and one of the chloroplast I have shown the inner structure. In the other chloroplast that I have drawn here as well as here, I have not shown the inner structure of that. Okay, so the plant cell has chloroplast that is unique to a plant cell only, that is present only in a plant cell. Now, the another structure that you can see in a big, huge structure, that is a vacuole. A plant stores its organic acids, its waste, some amount of water, all that is stored in a vacuole. And the vacuole has a fluid content. I hope you remember its name, that is called the cell sap. So, whatever fluid is present inside the vacuole, that is known as cell sap. So, that is present inside the vacuole. Okay, so plant cell also has mitochondria, but the number of mitochondria in plant cell is less as compared to that of animal cell, because the energy needs of a plant cell are less as compared to animal cell. So that is about the mitochondria. So that is all about the structure of plant cell and animal cell. I hope you are able to understand the structure and you are able to draw the diagram of plant cell and animal cell. That's all for this video. Keep on revising all the syllabus along with me. It's very important chapter for your future as well, because in 11th standard also you will be reading about all the cell organelles once again. So that's all for today. Good day everyone.